Hello, biology. Welcome back to another day. Happy Tuesday. Sorry I'm not there today to get you through this stuff. I'm still under the weather. You'll hear I'm a little raspy still. Miss you guys. Missing the week before Christmas break. It's one of my favorite weeks of the year, so I'm sad to be missing it with you guys. Today I'm gonna be going through the notes with you guys. We're gonna start on the fundamentals of genetics. Tomorrow we'll be doing Punnett squares. I hope I'm back tomorrow, but if not, you'll get another one of these. Uh, this is T, Brooklyn. I know you're worried about that, but we're gonna be talking about genetics. We're gonna be talking about Punnett squares. We're gonna be talking about how genetics are passed. If you need to pause this video at any point in time so everyone can get down the notes, please do so, and we'll just move through it. Just a normal day. All right, let's get started then. So, who was Gregor Mendel? Gregor Mendel was an Austrian monk. He worked in a monastery. He was not a scientist to begin with, believe it or not. He was actually a mathematician. In 1851, he entered the University of Vienna, and he was studying science and mathematics, primarily mathematics. His math classes were in a brand new, budding, state-of-the-art field of math called statistics. And this is actually like what was the key to him unlocking heredity and genes. So heredity is the transmission of characteristics from parent to offspring. This is why you look a little bit like your parents, but not exactly. You carry some of their characteristics, but not all of them. Is because of this passing from parent to offspring. So here's a picture of him in his younger years. Nice looking gentleman. So Mendel is famous for working with pea plants, like snap peas, kind of. And in the monastery, he was in charge of keeping track of these peas. And he noticed that these peas had different characteristics. Some of them were short, some of them were tall, some of them had wrinkly pea pods, some of them had smooth pea pods. And overall, he observed 7 different characteristics for pea plants. He called these a trait, and this is a genetically determined variant of a characteristic. And some of the traits he observed were pod color, the height of the plant, the flower color, the seed texture, etc. So these are what he was looking at, these noticeable physical traits that he could see on the peas, and he did his experiments with those traits. So in his experiments, Mendel would collect these seeds from these pea plants. He would tabulate, he would write down, record the traits and the seeds, and the results were not what you'd expect and not what Mendel expected. Short plants could grow from tall seeds, and white flowers could seemingly grow from purple parents. This was not what you'd expect. You'd expect purple plants to have purple kids, and white flowers to have white offspring. But this was not always the case. So he carefully and very meticulously controlled how these plants were pollinated. In fact, he would pollinate the plants himself, and he would take a paintbrush and he would take from one flower of one and then make sure that it went to the right plant that he wanted to pollinate. So in plants, pollination is a form of sexual reproduction when the pollen grains, which are in the male part of the flower, are transferred to the female part of the flower. And this is called the stigma. So the anther is where these pollens are made in the male reproductive part of the plant, and the stigma is where he would pollinate these, and this is the female reproductive parts of the plant. And some plants, most plants, have both stigma and anthers on the same plant. So you can self-pollinate them, you can pollinate other ones. It works out really nicely for in pollination season, like the bees like do the pollinating, but Mendel took it into his own hands. So there's 2 different types of pollination. There's self-pollination and there's cross-pollination. Self-pollination, here, one second, let me. Self-pollination occurs when pollen is transferred from the anthers of one flower to the stigma of the same flower or another flower on the same plant. So what this means is that DNA that we've talked about and that chromosomes that that plant has are going to be passed to that same plant. And in cross-pollination, it's taken from one plant and it's transferred to a different unique plant, different unique flower. Normally, pea plants self-pollinate, because of the proximity. Their flowers are really close to each other. Mendel, like I said, meticulously, like with a paintbrush, would manually pollinate these pea plants. And what this allowed him to do is that he could control the genetic flow. He basically decided who mated with who and which offspring were created. So in his experiments, Mendel, very meticulous, very good scientific method. He would look at one trait per time. So say, for example, the first one is he's going to look at the yellow pods versus the green pods. So in a true breeding plant, these plants with green pods, when they self-pollinated with themselves, always produced green pods every single time. In the example here, I go the opposite way. Plants with yellow pods would only produce offspring with yellow pods. So if I have a yellow pod tree, this yellow, it shows up. When this yellow pod crossed with itself, all the offspring would have these yellow pods. There'd be no green pods in the mix. And that was considered true breeding. So Gregor, let's say for that yellow and green pods, he made 14 true breeding plants, one for each of these traits. There were 7 traits. Each trait had 2 different types it could be. He made 14 of these plants true breeding. So every time they breeded with themselves, they created that green or yellow pod. And this was called the P generation. And think of it as the parental generation. He then would take the parental of one, like let's say trait, like let's say this yellow pod, and instead of matching it with another yellow pod, he then would match it with a true breeding green pod. So in both of these, normally if this yellow one had offspring with itself, it would form yellow, and if this green one had offspring with itself, it would form green. But he mixed and matched, so he took these 2 true breeding ones and he matched them together. In the F1 generation, which down here stands for filial, which is Latin for son or daughter, he would have the offspring, and he would record what their color was in this case. So in this case, where you had the yellow and green mixing, he recorded all the numbers of these and kept them very, very well organized. So this first generation is called the filial generation. So in the first one, we had in the P generation, let's do, sorry, I apologize. In the P generation, we had yellow mixed with green, and something I didn't tell you, and you should know, is all the offspring in this P generation were green. There was no yellow. The yellow disappeared. And then in the F2 generation, he took one of these plants, let's say it's this one right here, and he had it breed with itself. And so then he had 4 more offspring here, and the results is where it gets really interesting. When he had this true breeding yellow and this true breeding green, the 4 F1 were always 4 out of 4 of them were green. But then if he took this one of these, oops, sorry, got a little excited there. If he took one of these F1 generations and breed it with each other, so now we're going to go from F1 down to F2. What ended up happening is there was 3 green now, and my pods are becoming less pod shaped, I apologize, and 1 yellow. So he called these factors, the green and the yellow factors. We now call them traits. But what would happen is every time he'd have this 3 green to 1 yellow ratio. And this was for all his true breeding ones. Flowers, height, he always had this 3 to 1 ratio, and that's important. That 3 to 1 ratio gives us some clues as to what's coming up. So in his experiments, when the P generations were crossed, that yellow and that green, the yellow would disappear, it was covered by the green. We were calling that green trait dominant. So we're moving here, crossing those ones. This green was the only one. It was the dominant trait. Green equals dominant here. And the recessive was this yellow guy, the one that was hiding underneath of that dominant trait. So the dominant factor will always cover the recessive one, and if you have a recessive factor, it will always hide under the dominant one. They're kind of circular reasoning there, but that's how they work. The law of segregation, you remember when we had mitosis and meiosis, and we had our chromosomes lined up. And we'd always have them splitting off before they in meiosis and metaphase 1 and anaphase 1 of meiosis. These guys would separate to opposite ends of the cell. What ends up happening is each cell would get, excuse me, that was an awful line, I apologize profusely, would get one copy of each of these chromosomes. And when the two gametes would combine, they'd get two copies of each factor, one from each parent, one from mom, one from dad. Or if it's a self-pollinating plant, one from the plant and another one from the plant. Now, the law of segregation means that these factors segregated or separated during the formation of gametes. So up here in meiosis, when these are separating out, that is the law of segregation. These guys are separating out. Each gamete only gets one copy, and it's random which one. Independent assortment. This means that different traits are coded for by different factors. So a green pod can either be wrinkled, which is a different factor, or it can be round. Because it's green, it doesn't mean that it's a green pod's always round. It doesn't mean that a green pod's always wrinkled. Each one can be independently grabbed. So one chromosome could say, hey, we're going to be yellow, and then another one could say, hey, we're going to be wrinkled, even though wrinkled might be dominant and yellow might be recessive. So independent assortment means each trait, each of those 7 traits for those pea plants were randomly chosen. They weren't like, they don't have to be together. And remember, like in that last slide, we have those chromosomes splitting during anaphase 1 of meiosis. So independent assortment, the official textbook definition here, is the factors separate independently of one another during gamete formation, meaning that they're not inherited together, they're inherited all on their own. So support for Mendel's conclusions. Molecular genetics is the study of chromosomes and their genes. And a gene itself, this is an important definition, is a segment of DNA on a chromosome that controls a particular hereditary trait. So, for example, we talk about your gene for eye color, or your gene for height, or your gene for something called hitchhiker's thumb, where if your thumb is straight, you don't have it, but if your thumb like curls like this, like I'm like forcing mine back here, then you have what's called hitchhiker's thumb. The dominant trait in humans is to have a normal thumb. The recessive is to have that hitchhiker's thumb. So you can kind of like, and you're just like relax, like make a thumbs up. If your finger is more like that, you have hitchhiker's thumb, and if it's not, you have the dominant trait. And those different types of that trait are referred to as alleles. It's the different forms of a gene. So having the dominant allele means you don't have hitchhiker's thumb. Having the recessive allele means you do have hitchhiker's thumb. And this is what we call Mendel's factors today. So we can have an updated definition for them. Alleles are usually represented by letters. Sometimes they'll be related to like something that they do, like the one for, let's say, tallness of a pea plant might be T, and then a short one would be little t. So the dominant will be the capital letter, and the recessive will be the lowercase letter. So both of these code for plant height. Big T means tall plant. Little t means short height. Short plant. Short height. Excuse me. Alleles that are coded for the same gene get the same letter usually. So both of these are T, both of these are plant height. It's a good example. All right, we're going to do just that small chunk today. Tomorrow we're going to be talking about Punnett squares. You might have seen them before in your middle school career. We'll be talking about how like a tall plant that has a recessive gene and another tall plant that have a recessive gene can have both tall plants and this one short plant, which is this nice 3-to-1 ratio that we expect to see. So 3 tall to 1 short. Hopefully I'm back tomorrow, but if not, we will continue just plugging through this week. I miss you guys. Hope to be better soon. I hope to be back. Have a good rest of your day. Ciao, ciao.